Thank you so much for that introduction. Jeremy and I are very happy to be here today talking about our favorite topic, which is methanotrophic bacteria. I'm going to start out. I know many of you are not microbiologists and don't have a background. So I'm going to start out doing some pretty basic information about these organisms. And I apologize to those of you who are methanotroph experts. Then Jeremy and I are going to do a tag team, and I'm going to hand it off to him. And then I'll do some things at the end, and we'll hope for questions. Okay. So let me do this down here. Okay. So I just want to remind everyone, I'm sure you all know this, but regardless of the approach that we're going to use for methane removal to reduce atmospheric methane, there are at least two major challenges. The scale, which is huge, and I always advocate that we shouldn't be talking about an approach unless there's at least some idea of how we would get to scale. And I'm looking at, 10 to 15 megatons per year as sort of my idea. And then this incredibly low concentration in the atmosphere, which for any approach is a challenge, but for biology, it's especially a challenge because the methane has to dissolve in water. All right. So as we know, there are multiple approaches possible for methane removal. I personally think it's important to pursue many different kinds of approaches because we don't know right now which ones are actually going to be the most effective in terms of deployment. But biological methane removal is one of them. And there are some advantages for biological removal. One is that there are microbes that are known that will consume methane from air, so that the ability is there. They are called Methanotroph, which literally means methane eating. And these methane eating bacteria grow on methane as their sole carbon and energy source. So they do convert part of it to CO2, but they also convert a significant part to biomass, cells, basically. And it can be used as sustainable protein for feed, like in aquaculture. And there is a current market developing for that. So there's a co-benefit when you're using biology, and that is the biomass, at least if you can figure out a way to harvest the biomass. All right. Sorry. Okay. So what about methanotrophy in natural environments? Methanotrophs are very widespread in nature. They're found in virtually all environments where you have both methane and water. Biology needs water, all right? Places like lakes and wetlands and soils and oceans and estuaries and really all over. There are methanotrophs known that can grow in a range, very high pH range. Temperature range for biology is not terribly broad, but still you can get really low and you can get reasonably high. Now, there aren't individual strains that can use methane across the whole range. So you have some strains adapted to low, medium, high. But if you take the group as a whole, they are very diverse. And collectively, they serve as a biofilter to keep methane in the ecosystem. So I have this little diagram over here. Regardless of the source of methane in an environment, whether it's biotic, meaning biological, or abiotic, meaning not biological, that methane, when it encounters these organisms, can get oxidized to CO2 and biomass. Depending on the environment, you can get most of it oxidized or only part of it, but always a little bit leaks out. All right. So it's not a perfect biofilter in nature, but there is a great deal of the methane produced in nature that gets consumed, and it keeps that carbon in the ecosystem. Now, if you look at most natural environments, not every one, but most of them, this methane consumption is either limited by the concentration of methane or by oxygen. All right, so I'm going to go into where oxygen comes into this next. Right. So there are two major classes or types of methanotrophy, which is the ability to use methane as a sole carbon and energy source. There's a group of microbes that do this, they do it aerobically. They actually require oxygen to oxidize methane. Methane is converted in the first step to methanol with oxygen, right? And this, in general, these microbes that do this have a relatively high affinity for methane. And what that means is they have high activity, high ability to consume methane at low methane. So relatively high activity at low methane. There's a second group that are anaerobic. These are not bacteria, these are archaea. So that's a second domain of life, but they are microbes, they're single cells. Most of these are actually inhibited by oxygen. They need to be in environments where there isn't oxygen. The metabolism they carry out is very different from the aerobes. There are these bacteria that produce methane in nature. They're called methanogens, methane generating. And they have a special set of enzymes and metabolic pathways to make methane. What these anaerobic Methanotrophes do, this group, they reverse that methane production pathway. And the first intermediate is not methanol, it's a methyl derivative of a small cofactor called CoM. All right, and I'm not going to go into the pathways, but it is a relatively low affinity pathway. And that means it has relatively low activity at low methane. So if we are thinking about removing methane from gas streams where it's relatively low, and I'll take something like 1 to 2,000 ppm as a benchmark, these organisms are not really going to be great for this. In fact, we really want to look at the aerobes if we're going to be after this very low methane. Now, the anaerobes do consume a lot of methane in nature. They're in environments where the methane is high enough coming to them that they can consume it. So they're important environmentally, but if we are thinking about using very low methane, probably we need to be thinking about the aerobes. So let me talk a little bit then about this group of aerobic Methanotrophes. Again, they're using methane and oxygen, making CO2 and biomass. They have, again, relatively high affinity for methane. What's good about them as organisms to use is that the genetics is, relatively easy to do. Jeremy will probably wince when I say this, but there are effective systems that are available. We can do genetics in them. And what's important about that is that if you want to design in an organism, you change their DNA, and that means having genetic capabilities. And then genome sequences and omics databases, these system level databases that we use to understand organisms, are available for a reasonable number of strains. So as organisms, we know them fairly well, and we can work with them fairly well. So I'm going to now turn and tell you a little bit about this ability to oxidize methane, which is what separates these organisms out from other organisms. So for those of you who aren't biologists, when you see a word that ends in ASE, that's your cue that that's an enzyme. And an oxygenase is an enzyme that uses O2. A monooxygenase is an enzyme that inserts one oxygen atom into the substrate to make the product. All right, so methane monooxygenase converts methane to methanol, and it's abbreviated MMO. There are two versions of MMO that are known. Maybe there's more, but these are the two we know. One of them is found in membranes in the cells. It's called the PMMO. Compared to the other one, it has a relatively high affinity for methane. The other one is called the SMMO. It's found in the cytoplasm of cells, the inside. And compared to the PMMO, it has a relatively lower affinity. It's about a 10-fold difference. All right, so PMMO is a better enzyme, basically has a higher catalytic efficiency. These two enzymes each have a metal in their active site, so that's part of their catalytic mechanism. In PMMO, that metal is copper. All right, the SMMO, only a subset of these organisms contain SMMO. There are a few actually that only have SMMO. But most Methanotrophes that you might isolate would have PMMO. And it is an iron enzyme. All right, so once again, if you're going to be focused on using methane at these low levels, the PMMO is in the aerobic methanotrophes is the enzyme of interest, which makes copper very important to these organisms. So I'm going to hand this over to Jeremy now, because he's our expert in copper and methanotrophes, and he's going to give you some background in this.
Thank you, Mary. If we could just have the next slide, please. Mary talked about how copper plays an important role in one of the two forms of the methane monooxygenase, what we call the particulate methane monooxygenase, which is found in the membranes. What I want to show to you is that copper is actually a key component or a key parameter that we have to consider if we're going to be trying to utilize methanotrophes for the removal of methane, particularly at low concentrations. And here what I have plotted on the y-axis is the activity of the methane monooxygenase versus the copper concentration that we grew the specific methanotroph in. And what I want to highlight in this particular slide is if you grow the cells in the absence of copper, there is a certain amount of activity. And according to this assay, it's just under 200. I don't want to go into the units too much, but just keep that value in mind. It's about 200. But all of the activity is found in the soluble fraction of the cells, that is in the cytoplasm, and that's due to the soluble form of the methane monooxygenase. If you add just as little as 1 micromolar copper, all of that activity disappears in the soluble fraction and it starts to appear in the membrane fraction. That is, you're actually converting methane from using the soluble methane monooxygenase in the absence of copper to the particulate methane monooxygenase in the presence of copper. And what I want to stress here is the activity in the membrane fraction goes up, continues to go up as you add more copper to these cultures. And more importantly, you also see that in the whole cell. In the whole cell, we see a certain amount of activity in the absence of copper, around 200. But if I add in, say, between 20 and 30 micromolar copper, I can actually increase that activity by about 50%. And so the availability of copper is critical for these cells to be able to oxidize methane. And it's critical to consider if we're going to be utilizing these microorganisms to remove methane or to control methane emissions from various environments. And so I got curious. And I went along with one of my colleagues, Alan DeSpirito, wanted to know how were the cells collecting copper. And so could I have the next slide, please, Mary? Now, this may seem to be a bit of a detour, but I'm going to take you back on the right path just a minute. But I have to make sure that we're all on the same page and understand why it's important to know how methanotrophes collect copper. Because we know that the methanotrophes sense copper. They find it in the environment, okay? And that causes the relative expression of the two forms of the methane monooxygenase to change. But there's a lot of variety of copper speciation in the environment. It could certainly be free, existing as copper-2, but it could be associated with organic matters, or it could be absorbed onto soils. How do the cells find this copper? I mean, what mechanism are they using? And I'm just giving you the thought process that we went through. And at this time, at this level of analysis, we didn't know whether these cells were utilizing a copper-specific system to find copper and pull it inside itself, or was it a happy accident where there was another metal uptake system that would also take up copper for these cells? And so Alan DeSpirito and I wanted to know more about what was going on, what was causing or enabling these cells to collect copper. Okay, so could I have the next slide, please? And what Alan and I found was, if you grow methanotrophes in the presence of a little bit of copper, not the absolute absence of copper, but just a little bit of copper, if you grew them up in a little bit of copper, in this case about 0.7 micromolar, I don't know why it's doing that.
I don't know why it's doing that.
If you grow it in 0.7 micromolar copper, if you grow the cells up and then spin the cells down and collect the spent medium or the supernatant, as you see, as I hope you can see on the left, is a bright yellow. Okay, but if we grow the same methanotroph in the presence of elevated copper, 10 micromolar copper, that yellow coloration went away. And Alan and I were very puzzled by this, but we speculated. We thought, well, what if we just made a hypothesis? What if at 0.7 micromolar copper, there's too much copper for the cells to be able to express and utilize the soluble form of the methane monooxygenase to oxidize methane, but there's not enough to have high activity of the particulate methane monooxygenase? What if these cells are stressed for copper and they're spitting something out to try to find more copper and pull it inside themselves? And in the presence of 10 micromolar copper, they don't need to make this compound because there's lots of copper available to them, okay? And long story short, Yes, the cells are spitting out a specific copper binding compound, if I could have the next slide, please, that we call methanobactin. This is a really interesting compound because it's a modified polypeptide. If you look closely, and if you know your biochemistry, you can see that it has several amino acids associated with it, but it also has these two really weird heterocyclic rings. I don't know why it's doing that, but
I don't
sorry,
know why.
guys.
Sorry.
It has these
My
heterocyclic
computer
rings.
somehow.
Yeah, it has these heterocyclic rings. And if you look closely at those rings that I've labeled A and B, there's that nitrogen sulfur groups associated with those rings. It's those nitrogen sulfur groups that bind copper extremely strongly. In fact, this particular compound that we call methanobactin has one of the highest reported affinities for copper ever reported. Okay. In fact, this has such a high affinity for copper, it can actually dissolve copper-bearing minerals. All right. And so when we deduced the structure of this compound and figured out that it bound copper so strongly, we wanted to know if this is the mechanism whereby Methanotroph collect copper for their activity, but by doing so, could they inhibit the activity of other microorganisms? And this is where we get back into the story of how this is important for greenhouse gas emissions. If I could have the next slide. Methanotrophes aren't the only group of microorganisms that require copper for their activity. Another classical group of microorganisms that require copper are also the denitrifiers. And the denitrifiers don't utilize oxygen as their terminal electron acceptor. Instead, they utilize nitrate. And what they do is, in the absence of oxygen, these microorganisms will take and dump electrons to nitrate, first converting it to nitrite, or NO2 minus, further reducing it to nitric oxide, or NO, then reducing it even more to N2O, or nitrous oxide, which we all know is a very potent greenhouse gas. And then finally, they will convert the nitrous oxide and further reduce it to dinitrogen. That's the complete denitrification pathway. And it turns out that that last step, if you could just highlight that last step for me, Mary, that last step, it's mediated by an enzyme called the nitrous oxide reductase, or NOZ. That enzyme has a very high requirement for copper. In fact, there are 12 coppers per protein. Okay, we speculated that if microorganisms that denitrify didn't have copper, would nitrous oxide reductase activity be inhibited? Would denitrification stop at the level of nitrous oxide? And fast answer, yes. Could I have the next slide, please, Mary? In this case, what we did is we did a very simple study where we took 3 cultures of known denitrifiers, okay? And we just incubated them in the absence and presence of methanobactin. The absence is on the top and the presence is down below. And for all 3 of these denitrifiers, if we incubated them in the absence of methanobactin and then chased the various intermediates in the denitrification pathway, what we see is that nitrate is all consumed. Those are the squares. And you occasionally see a tiny little blip of nitrous oxide, okay? What this is telling you is that as fast as nitrous oxide is being made by these microorganisms, it's being converted into dinitrogen. So there's no buildup of nitrous oxide in these systems. But if we actually add in 1 micromolar methanobactin, okay, and that's not a lot of methanobactin because methanotrophes can actually make quite a lot of it. If you add in just 1 micromolar of methanobactin, all of the nitrate is stoichiometrically converted into nitrous oxide. That is, if you look for the first one, dichloromonoser aromatica, RCB, you see the nitrate go down, but the nitrous oxide goes up, and it's a 1-to-1 ratio. That is, the denitrification pathway stops at nitrous oxide. The cells cannot complete denitrification. And so what this tells us is that, yes, there can be copper competition between microorganisms. And if you are interested in stimulating methanotrophy to actually increase methane consumption and to reduce methane emissions, if you're not careful about it, you could actually starve other important microorganisms for copper and make the problem worse, because as we know, nitrous oxide is a much more potent greenhouse gas than methane. Now, this was done using a pure culture study approach. If I could have the next slide. We also showed that it happens in soils as well, okay? In this case, we took these are 3 different rice paddy soils that had different natural abundances of copper associated with them, and we incubated them in the presence and absence of methanobactin. In the absence on the left-hand side, for all of the soils, there was very little, if any, nitrous oxide accumulation. It was a tiny blip, okay? But if we added methanobactin, we did see accumulation of nitrous oxide, okay? But what I want to point out is if you compare the nitrous oxide profiles in soil A at the top versus soil C at the bottom, you'll see a different pattern. In soil A, there was permanent accumulation of nitrous oxide, okay? But that soil had about 1 milligram of copper per kilogram of soil. Soil C, you saw some initial production of nitrous oxide, but then it slowly decayed away. That soil had a much higher copper content, about roughly 5.75 milligrams of copper per kilogram soil. And what we speculate here is what is happening is that initially when we added the methanobactin, it bound all the readily available copper, limited copper uptake by the denitrifiers present in this system, but over time, copper re-equilibrated, copper dissociated from the soils. It was reintroduced into the aqueous environment, and the cells were then able to take that up, and then subsequently were able to reinvigorate the nitrous oxide and then remove the nitrous oxide via the nitrous oxide reductase. So what I want to stress is, you can have a problem if you're uncareful or unwise in your approach to stimulate methanotrophes. You can actually induce greater nitrous oxide emissions. That's a big problem, but there can be a simple solution just by making sure that there's enough copper to go around for all the members of the microbial community. Okay, so if I could have the last slide, the summary slide for what we're going to talk about here. Just want to reemphasize the implications of copper and how it impacts methanotrophy. There's clearly a link between the methane and nitrogen cycles, and that link is coordinated by copper. And so we have to think about copper if we're going to be utilizing methanotrophes for methane removal, because we don't want to inadvertently increase nitrous oxide emissions. But how best to do this is how much copper should we add, okay? That's something that we're doing right now. We're extending these studies where we're looking at various soils, both agricultural soils and prairie soils, and asking ourselves is how much copper should be added to these environments if we're going to try to ensure that we can both remove methane while preventing nitrous oxide emissions. Okay, so that's my little snippet with respect to methanotrophy for this presentation, and I'd be happy to answer any questions that you might have at the end. And now I'll pass it back to Mary.
Okay, thanks, Jeremy. So this is why we wanted Jeremy to go through the copper story, because it turns out to be incredibly important if we're going to talk about, as a community, environmental manipulation of methanotrophy to increase methane consumption. So it's really important to this is a possibility. I think we need to consider it, but there are some unknowns that Jeremy and I think is very important for people to think about. So we need to understand these ancillary impacts, as Jeremy just said, including N2O production, because you have very complex communities in these environments that might be manipulated and really need to be measuring a little bit more than just methane. And if you go into the environment and you look at these, go into the literature and look at these papers about stimulating methanotrophy and reducing methane, it's very rare that they measure N2O. So it's really important that both be measured. N2O is a little hard to measure, actually. So that's part of the problem, but it's really important. All right, another issue is in many of these environments, not all of them, but in many of them, the amount of methanotrophy is methane limited. All right, so we don't want to add methane, obviously, to stimulate it. So that's going to be an issue. It's unlikely, as Jeremy pointed out, that copper is going to be a problem because they have this amazing copper sequestration mechanism. Now, a subset of this is in landfills. It's becoming, relatively common to put a biocover on it that's very porous. It allows oxygen diffusion in and stimulates this aerobic methane consumption. But you don't really find these studies measuring N2O as well as methane. And it could result in N2O increase because these covers are usually not going to have a lot of copper in them. So it just depends on what community forms there. And then, as in all of these environmental manipulations, how are you going to reach the scale of tens of megatons a year when the required land area is so vast? That's just that, not just, that's a scale up issue. All right, so environmental manipulation of methanotrophes is certainly possible, but I think still needs quite a bit of work. All right, so I want to take just a minute or two to talk about how much methane is in the gas stream you're treating versus the treatment options. And there are categories, rough categories. If your methane is more than 20%, theoretically, at least, it can be flared or used for energy. It's not efficient enough at 20%. Obviously, even at 50%, I know it's problematic. But there's technology that could possibly be optimized. Once you get much below 20% in that 1 to 20%, actually, there's existing chemical or biological technology that works. It may not be economically feasible. It needs to be optimized, but that's easier than developing something brand new. Once you get down significantly below 1%, and 0.02% methane is 200 parts per million, then we really need new technology if we're going to use methanotrophic removal. We have strains, but the current kind of process technology just isn't really up to these lower levels, especially as you get down to 500 ppm and below. And once you get significantly below 200 ppm, then we not only need new technology, but we probably need better strains. And that's what we need to get at wetlands and ambient air. All right, so I always think immediately about what's the concentration that's the target. And that then tells you about what the different options might be. Okay, so what we're looking at is to target this 100 to 500 ppm methane in air in a closed system, not out in the open environment, because there are all these issues, but a closed bioreactor type system. Jeremy's group published a paper in 2009 where they modeled the use of low methane. And he showed, and we've shown in our own lab that, and others have, that aerobic methanotrophes, many of them can stably consume methane at 500 ppm, and they will remove it down below 10 ppm. That you can roughly estimate that there are on the order of tens of thousands of sites in the US alone. There are emission sites where the air above those sites does have elevated methane in this sort of 100 to 500 ppm. So, you know, these are sort of the common emission sites you can imagine. And so you can, you know, do a back of the envelope calculation and say, actually, the scale is there using a closed system in terms of the amount of methane at this level. What methanotrophes would we use? It turns out that many methanotrophes actually can't grow at 1,000 ppm and lower. They're really, really low for them to grow. If you want them to be saturated and maximum, you're usually going to use at least 5%, which again is 50,000 ppm. So what we did in our lab is we screened a suite of methanotrophes that we had, and we found most of them won't grow at 1,000, but we found a small subset that would grow at 500. Or so 1,000 and below. And so here's some data. What we're looking at is growth. As these values increase over time, that means the number of cells is increasing and they're growing. And these are all different strains that we tested. These are all strains that grow a little. We had many that just don't grow at all. What you can see is that some are pretty good at this and some are really poor. And down at the right-hand side is this is just a measure of their growth over time. And the one that is the best is this M. burriotense 5GB1 strain, which is a strain we have studied a lot and work with a great deal. We were a little surprised that this one that grows really well at high methane would do so well at low. But this is a strain now that we're beginning to work with for this purpose. And in this strain, but also we've tested it with some of the other ones that will grow at on the order of 1,000 ppm. Once you get below about 2,000 ppm, what we're looking at here is the amount of methane uptake per gram of cells. versus the initial substrate concentration. And there is a linear correlation with concentration for the rate. So it does mean that probably most of the time we can extrapolate rates to lower concentrations if we have measurements that have been made like below 2000. All right. So here's the concept that we have, this sort of closed system bioreactor approach with aerobic methanotrophes. We target these emission sites where there are high local concentrations of methane in the air, high meaning 100 to 500 ppm. You're going to have some kind of blower or pump. And we're envisioning a shipping container size, portable, low-cost system where you're growing these methane utilizing communities. We're envisioning this being modular, so it could scale relatively easily. It's got to be simple and low cost and low power. And then you will be making a product. Remember, this is the co-benefit. Sustainable protein is one possibility. You just take the cells and dry them down and they're naturally half protein. And so, you know, these have been used again for animal and fish food for quite some time, or they could be engineered to make another high value product, fuels or chemicals. That's going to get a little more complicated. But you have this product that if you can figure out how to harvest it inexpensively and effectively, will help drive the economics. All right, and roughly what you would get for every ton of methane used is 1.25 tons of CO2 and about 0.8 tons of this biomass. So you get a reasonable amount per ton of methane. Okay, so how about scale? You know, how are we going to get to this 15 megatons a year? There is existing biofilter technology in the waste treatment literature. They're called trickling filter systems. They are mostly designed for 1% and higher methane in gas streams. And there are really 2 studies that have looked below 1%. And they have really low daily consumption as you get down to these much lower levels. Again, Jeremy's group, here's this paper again, and there's another study that's been done a little bit later, that if you looked at a sort of standard methane removal unit size, 120 cubic meters, the estimate is it could remove on the order of 10 tons a year if you had 500 ppm in the gas stream. All right, so, you know, a little bit of multiplication tells you, whoa, you'd need 1.5 million units worldwide to consume 15 megatons a year. There aren't that many sites probably. All right, so this technology as it exists isn't going to get us to scale, much less to economic feasibility. So there's a target then. We need to increase the annual consumption per unit, which is the capacity of these treatment systems. If we could get it up tenfold on average, we'd need something like 150,000 sites globally. But if we could get it up 25-fold, we'd need 60,000 sites. This is a lot, but the sites are there globally. So we know the volume of methane is there. It would be very challenging, but at least it gives us an idea of what it would take to get to scale. Okay, so we got to increase the capacity, right? This methane consumed per cubic meter of treatment volume per year. We'd like to increase it 10 to 25-fold. These current trickling filter systems aren't designed at all for using low methane, and the mass transfer is a real issue. So I actually think there's probably a lot of possibility for improvements on this capacity number for the bioreactor. There are a lot of groups now working on thin film bioreactors that really increases the mass transfer, and then getting increased numbers of cells per cubic meter in your system, and then working on efficiencies as you scale and efficiencies for energy. These are all areas that have to be developed. And then improving the culture. I'm optimistically hoping we could get 5 to 10-fold. We're working on this right now. We want higher methane consumption per cell at these low methane levels. We want a simplified, optimized culture medium We're testing now about the nutrients that are needed. Jeremy pointed out the copper, and copper optimization is going to be really important. And then we need to ensure no N2O production. We can do that, of course, using strains that don't have the ability to make N2O. And the ones we're working with right now don't. Okay, so now let's get really challenging. Could we target ambient methane, like near ambient? Let's say the 2 to 10 ppm level. You know, wetlands probably, you know, a lot of them are in that 10 ppm. And I'm sure, you know, all of you are very aware of this sort of scary issue that as the earth warms, the methane for wetlands is going up. And we don't really have a good solution for wetlands. But what you can do is estimate, well, what if you had one of these closed system biofilters, like we're thinking about for 100 to 500 ppm, could you get down to 2 to 10? You have to increase the current elimination capacity 50-fold. Very challenging, but at least it's a target, right? How would we do it? Well, we could engineer the strains, and we have some ideas and a couple collaborators, and we're starting to work on this. Whether it'll work or not is still, you know, it's very high risk. And then we'll need some novel bioreactor configurations, probably to concentrate the methane, as well as increase mass transfer. So that's sort of out in the future, but something that at least is theoretically possible. All right, so that's where we were with our prepared remarks. The summary is that there is a potential to use biological methane consumption to contribute to the issue. We have the tools and strains and data sets and expertise to work with these organisms. Manipulating Methanotroph in natural environment, we've got to address this N2O issue. But clearly, if we're going to get to scale and use Methanotroph, we're going to need advances in the biology and the reactor engineering, that probably economics and policy as well. So encouraging signs, but we're not quite there yet. And then some acknowledgements for both of us, for our collaborators and our funders. And I think we'll stop here and ask for questions.